Okay, in this video we will talk about the Angiosperm family of Lamiaceae. The data is here, Lamiaceae. So I did, of course, some cleaning in this data, and I'm gonna go through it here. And of course, you know this very well, you did yours. So you have the species here, you have the leaf length, leaf width, area. I calculated the area there, the plant height, and RBCL sick and mat sick. So I uploaded this. You can upload that from here. Upload. So you have the data there in Excel. Now here we will do some exploratory data analysis. So of course you inspect your data as we just showed in the other video. You do some data clean. Then you can run some exploratory data as we will do. Same thing, we import the libraries that we will work on. We have pandas, numpy, matplotlib, seaborn, scipy to do some stat, and scikit-learn actually is to do some machine learning, but I will not get to that here. So here is in Excel. You see in the other one with CSV. Now we can do Excel by using read_Excel and Lamiaceae data. So I run this, then run the data. My data is, then I can see my data. You see here, now you see just like the Excel. Of course, this index is added by Python. You have the species here. Okay, I did 10 here, so we can see 10 from 0 to 9. You have leaf length, leaf width, area, and plant height, and you see the RBCL and MAK sequences there. Of course, these are categorical data now, and these are numeric. So then what I did, basic overview, what are the data type, and you see if you have any missing data, but here I made sure there are no missing data there. And describe will give you the stat, and you round that to 2 decimals instead of 4, 5, or 6. So here it tells you that leaf length is a float, it's a number, all these are float, plant height is an integer, RBCL is an object. So there are no missing data here, everything is 0, so every single cell has a number there, which is good. And you have some basic statistics as we mentioned earlier. We have only 50 counts. You see you have your 50 plants. Of course, only 10 showed here, but that's how Python does things. Unique species 50, then you have the count is 50, mean for leaf length 8, standard deviation minimum 20, quartile 58 and 75, then maximum for all your data. Then here run this to handle missing data, but we don't have any missing data in that. That's good. So then you do same thing, we run this, and we have the isoflavones. You have here a distribution of leaf length in your population, and here leaf width, leaf area, and plant height. Of course, these plants are not highly related like the rails. The rails, how we do them, we cross 2 parents, we have F1, we cross F1, we go until F6. So all these are actually related. They are recombinant inbred line, and the distribution is more or less normal. Here you just collected different species, although they belong to the same family, Lamiaceae here, but you might not have a very good normal distribution of your trait, which is leaf length here or whatever. But it's still a little bit normal here, at least leaf length, leaf width. Also, this is skewed a little bit, this one also. Univariate analysis, if I did that is gonna be the same. Then running this code will give us the same as we did for isoflavones. You have these pairwise scatter plot. So if you see leaf length here with all the 3 others, leaf width, area, and height, there is a correlation there. Here not so much with plant height, but at least here. Same thing, leaf width is correlated, but not plant height with the other 2. Same thing here, leaf area, but not here. And plant height is not correlated with anything. So you have here the heat map with the correlation or correlation map. So close to 1 is highly correlated, of course, and very low correlation here with the plant height. Then I did this to actually save these, so they are already saved there. I will get to that. Then I ran this to give us the species. You have the species names here. It's called group analysis. Then it will give you the leaf area, leaf width, and all the other. So of course this is to make it to flip it. So you run this here and you can flip them, which is nice here that you can see the species in here vertically. Then you have leaf width or whatever is there. You can see some groups. This group of plants have similar plant height, some of them have high plant height, etc. Then we do the summary as we did before. That's just summary statistic, and you save it. This is will save. Now when you run these, you will have actually these, so you see them all here. So if I click the bivariate, all the graphs are here. I can click on this, you see the graph heat map there, you see the other graph we went with. It saves these there, and you can download them and use them in your paper or abstract or whatever, okay? Leaf width, all these are there, individual and group. So that's basically some exploratory data analysis just by doing the numerical ones. Okay, I did not do or start to do anything yet with the RBCL. So now I will show you an abstract. So I wrote this for you. It's going to be study of several morphological traits in the Angiosperms family, Orchidaceae or whatever is your family. Here it's Lamiaceae, your name and mine, my affiliation, my lab and the department. So here I want you to write a small paragraph about the evolution of Angiosperm. You see here the Angiosperm Lamiaceae family, commonly known as the mint family, is one of the largest families of flowering plants, encompassing over 7,000 species spread across approximately 236 genera. This family is well known for its aromatic members, many of which produce essential oils, making them valuable in culinary, medicinal, and ornamental applications. Species like lavandula, lavender, mentha, mint, salvia, sage, and rosemary are widely cultivated and appreciated for their distinct fragrance and flavors as well as their therapeutic property. That's a very good intro. Then you state your objective of the study. So you see here, the objective of this study were to describe several morphological traits such as leaf length, leaf width, leaf area, and plant height in 50 randomly selected species of the family. Then the results showed that the leaf area distribution appears roughly normal, etc. Then I put here, you write something with these. You see interpretation here with your graph plot with some description, etc., heat map, and these other figures there and some references, etc. I'm still working on that. Then from that you write the main result. Okay, so here for example it shows the results showed that the leaf area distribution appears roughly normal, leaf width distribution is slightly right skewed, the leaf area distribution is heavily right skewed, and the plant height distribution shows a moderate right skew. In this sample of Lamiaceae species, distinct morphology in terms of leaf size and plant height, revealing diverse adaptations within the family. Species such as Salvia, Salvinia, etc. exhibit large leaf areas. You will see that from this graph, driven by both longer and wider leaves, suggesting adaptations that may support a higher photosynthetic capacity. Conversely, smaller leaves species like Thymus, rosemary maintain compact leaf structures potentially as adaptation to drier or nutrient poor environment, because you will see in literature, and they will get to that, that the more plants have broader leaves, etc., you have high photosynthetic rate. So overall, this morphological diversity highlights the adaptability of Lamiaceae species to different ecological niches, with variations in leaf size and plant height that reflect a range of growth strategies and environmental adaptation. That's your conclusion there. So with that, that's your abstract, and you can send it to a conference, etc. Now you do the abstract first. Before you do the abstract, of course, you will do some writing and interpreting of your results as I showed you here. Then now from this you can do your poster.